water and oxygen. So in the catalyst test, how it's performed is that a slide is taken, a colony of bacteria is placed on it, okay, or emulsified or not pun boleh, and then you put a drop or two of hydrogen peroxide and you see what? Bubbles will form. Bubbles will be forming. You can do this in your next practical lah. Okay, whereas so this is the first test besides gram staining that can differentiate between Staphylococcus and Streptococcus. So all Staphylococcus produce catalase, Streptococcus does not. Okay. So three Staphylococcus that we will look at that are important lah. There are only three that we will look at will be aureus, which is most important, epidermidis and saprophyticus. So this is a general chart showing how these three can be differentiated. So as I said, they are all catalase positive. Okay, Staphylococcus are all catalase positive. But aureus is also coagulase positive and mannitol positive. Okay, mannitol is a sugar, so Staphylococcus is able to ferment it lah. And we'll see that plate later. And coagulase is the ability, I think we saw coagulase kan in our pathogenicity lecture. So some bacteria can produce coagulase where they coagulate or do the clotting, right? So Staph aureus is coagulase positive, whereas the rest are not. Okay, so this is one image I took from this book called Clinical Microbiology, made ridiculously simple. If you have seen it in the library. So this image here was supposed to help you remember lah what Staphylococcus aureus is. Okay, so what do you see in this image? Yeah? What do you see in this image? A group of
people.
A group of this is actually supposed to be hospital staff lah. Okay. Because staff, we always say staff, but in exam of course you must write in full lah. Okay, but in short they call it staff. Okay, so the hospital staff all coagulating together lah, according if you read the book it describes that way. So because Staphylococcus aureus is often nosocomial pathogen, okay, meaning hospital acquired lah, okay, often transmitted in the hospital, okay, and then they are all wearing what? Gold medal lah, because aureus means gold, and on blood agar plate they produce typically they should produce yellowish colonies, but sometimes when we grow organism again and again in the lab for storage in the they lose certain properties, so tak nampak lah the yellow. Okay, but usually clinical strains you will see they are very yellow, and you can see the color is yellow lah, bright yellowish at least. Even last practical, if you notice, the single colonies were a bit yellowish. Okay, the rest were greyish or whitish. Okay, what else do you see? Cats lah, that's for catalase positive. Okay, so it's just simple diagrams that can help you remember some because your microbiology mostly, especially the first term, I think is all about remembering kan, banyak a lot to memorize. Okay, and one more thing is most isolates produce beta-lactamase. What does beta-lactamase do? They bind to beta-lactam kan, beta-lactam antibiotic, so they bind to penicillin. So they are called penicillin binding protein. Remember we saw. So because they produced beta lactam means they can be resistant to penicillin. A lot of them. Okay. Okay, so it's one of the most common cause of human infection. So you will see them a lot in your even in your systemic infection. Okay. Okay, so the diseases they cause can be divided into inflammatory diseases and toxin mediated diseases. Okay, so inflammatory including skin infections, impetigo, cellulitis, abscess, okay, furuncles, carbuncles, wound infections. So you would see this in the skin lecture. They can cause acute endocarditis, septic arthritis, osteomyelitis, right? Hospital-acquired pneumonia, meningitis, cerebritis, brain abscess, and also blood and catheter infection. So they can um, actually affect many organs, okay, through various mode of entry, right? So that was the inflammatory ones, and toxin mediated there are three. Okay, gastroenteritis, um, or food poisoning. Okay, so in food preparation, if the stuff if the food is contaminated with staphylococcus that produces exotoxin, produces exotoxin that is it can cause food poisoning. Okay, toxic shock syndrome and scalded skin syndrome. Okay, so this is again some images emphasizing on those. Okay, and most common causes of hospital acquired pneumonia, septicemia, and surgical wound infection. Okay, so from the nosocomial lah. Okay, nosocomial infection. So even studies have shown necktie, necktie pun when the doctor wears a necktie, they see one patient to another. Okay, through that also it has transmitted. Okay, because they maybe need to bend down to look at examine patient. So that's in the past lah. I'm sure there's a lot of measures are taken. You tuck in your tie neatly kan into your lab coat and so on. And hand hygiene is especially important to avoid this. Okay, from doctor seeing patient to patient, not to transmit it from one patient to another. So how is it transmitted? Okay, so generally all organisms, when you learn your first semester, you go organism by organism. You learn when you do when you want to part on transmission, you must think of the habitat. Okay, from where? Okay, and how it's transmitted. So Staphylococcus aureus is mainly present in skin, okay, and in the nasopharynx. Okay, so transmission is through contaminated hands or through ingestion. Okay, so it can when you touch your nose, people who are carriers, you can then don't wash your hands, you can transmit it into food that is being prepared or to another person when you touch. Okay. It is an opportunistic pathogen that can cause variety of self-limiting to life-threatening disease. Self-limiting meaning it can heal by itself lah, kan? Or it can go up to life-threatening. Okay, so through inflammation and toxin. The virulence factors are the structural defenses, enzymes and toxins. So I'm not going to repeat all this. Okay, we have seen this in the pathogenicity lecture, but it's important for you to remember lah what Staph aureus has. Okay, so it can have capsule, okay, protein A, coagulase, bacterioglycan teichoic acids. Coagulase, as I mentioned earlier, okay, converts the blood fibrinogen into fibrin to form a clot. Okay, so just now we saw only aureus produces coagulase kan, as compared to epidermidis and saprophyticus, right? There are three kan Staphylococcus. Okay, so only coagulase, only aureus among these three species produces coagulase. Okay, enzymes. Okay, so besides coagulase, it has fibrinolysin, hyaluronidase, lipase, and beta-lactamase, also called penicillinase. I don't repeat that. Toxins. Okay, so have you heard of super antigen exotoxins? Super antigen toxin. Dia kan masa immuno, I think. So super antigen exotoxin ni, why they are called super antigen? Because okay, if antigen can elicit immune response, super antigen they elicit a more general immune response. Okay, so they have an affinity for the T cell receptor, MHC class 2 complex. They have not a specific immune response but a general one. Okay, so the enhanced T lymphocyte response activates large number of helper T cells, causing overproduction of all the inflammatory cytokines. Okay, IL-1, IL-2, IL gamma, TNF. So the inflammation that is caused is from the immune response lah. Okay, so that's why it's called super antigen. So it has two types of super antigen: enterotoxins and what is TSST? Toxic shock syndrome toxin 1. Entero, from the name, it means what? Gut lah. Entero means gut related lah. Okay, so this enterotoxin causes gastroenteritis. So when ingested as preformed toxin, so what is meant by preformed toxin? Preformed meaning it's a toxin that has been released lah. Okay, it's not part of the bacteria but it is released maybe during preparation of food, okay, or food that is already contaminated. Okay, and even a small amount of the toxin can induce symptoms of staphylococcal food poisoning within few hours, and it is heat stable. So even cooking can still cause gastroenteritis. Okay, so that's why even storage of raw food, meat, or after cooking, storage after cooking is so important to refrigerate. Okay, because you don't want the bacteria to proliferate and start producing toxin. Okay, there may be bacteria there that can be killed through in a small amount, but it can be killed through heating. But once it sits on your bench top in your kitchen counter long, or you have overthought, you can the bacteria can start growing, produce toxin. So the toxin will stay even if you have cooked the food. The bacteria can be killed, but the toxin can still cause food poisoning. And this enterotoxin, it has a short incubation period. Okay, so when an organism has a short incubation period, an organism that causes food poisoning, it usually causes vomiting lah. Okay, so immediate effect. Longer incubation period might cause diarrhea. Okay, but sometimes it's not always the case. Yeah. Okay. Okay, but usually. Okay. So it's you will see these organisms again when you do food gastrointestinal and foodborne, right? But this semester focus is more on how you differentiate bacteria, okay, how you identify them, the pathogenicity, the toxins that are produced, because you won't go into those details next semester. It's more of looking at the whole systemic infection as a whole lah, and then you do see what are the organisms. So this part is important to remember lah. Okay, what are the top types of toxins and all. Okay, so next, toxic shock syndrome. Toxin 1 causes toxic shock syndrome, produced locally by Staph aureus in the vagina, nose, or other infected sites. It's often related to the use of tampons lah. I don't know if the guys know what tampon is, but it's a form of sanitary napkin that is placed inside the vagina. Okay, so that the Western culture they don't want to cause it's able to absorb and stay longer lah. Okay, so in which case no need to change sanitary pads frequently. So but then it caused there was a big epidemic whereby the use of super absorbent tampons by menstruating women they seem to get this toxic shock syndrome, because what happens, the bacteria in the vagina, which is where its habitat is, it starts to proliferate, because it has a when the blood is like what do you say, there's no movement lah, there's a nice environment for it to grow. Okay, there's collected blood that's not removed, by right the sanitary napkins should be changed frequently kan. So this one allows longer absorbance and so-called super absorbent tampon. So bacteria is so allowed to grow. So when they grow, they start producing toxin, and the toxin can cause toxic shock lah. Okay, it can also happen in the nasal packing used to stop bleeding from the nose. Allowed for use very long. Okay, so it can cause fever, nausea, vomiting, watery non-bloody diarrhea, rash similar to scarlet fever, and septic shock and organ damage. So that was the super antigen exotoxin. Okay, the next toxin produced by Staph aureus is the exfoliative toxin that can cause scalded skin syndrome in young children, causing this slowing off of the skin like kulit lupas, but this is not its mortality due to it is low lah. Okay, this heals. Okay, so there's two kinds of epidermolytic toxins involved, toxin A and B. The acts as protease that dissolves the epidermal desmosome. Okay, so it breaks the epidermis causing skin peeling. So usually affects neonates with local infection of the recently severed umbilicus or older children with skin infections. Okay, so that's why in surgical procedures, even the umbilicus removal must be done with using sterile equipment, okay, to avoid infection. Okay, besides that, it produces cytolytic exotoxins. Okay, so this causes disruption of membrane, including red blood cells and leukocytes. So you have the alpha toxin, penton valentine leukocidin. Okay, this causes destruction of white blood cells, and the alpha toxin of hemolysis lah. Okay, red blood cell. Okay. So how is Staphylococcus aureus diagnosed? Okay, so all those that I mentioned earlier, catalase positive, coagulase positive, and it ferments mannitol. So mannitol, this is a mannitol salt agar. where it shows that Staphylococcus epidermidis here is grown along with Staphylococcus aureus on the other half. So Staph aureus ferments mannitol. Okay, so um, it also contains, this agar also contains salt lah, so it's called mannitol salt agar. So it inhibits other bacteria. Okay, so because Staphylococcus it can grow on salt with 7.5% salt, it can still grow. Most other bacteria cannot. Okay, um, and between these two, you can use the agar plate to differentiate lah epidermidis from aureus from the other stem. Okay, and then these are the yellow colonies I was telling you about on blood agar plate. And then it also produces beta hemolysis. We could see this the other day on the practical. One of the plates had Staphylococcus aureus kan, 8M or something. Okay, so it was beta hemolytic. I will show you more plates on that later. And antibiotic susceptibility test is important. Okay, because many strains are penicillin resistant. Okay. Okay, this is Alexander Fleming. Have you heard of Alexander Fleming? What did he? Okay, I already speak that. He discovered
Penicillin.
penicillin in 1928. Do you know how he discovered?
Fungus. Ada fungus. Penicillium something.
Do you know how he discovered penicillin?
By accident.
So Fleming is a scientist kan, who was known to be very untidy. So the hikmah behind his untidiness is that he when he went off for a holiday, he left the plates lying on the bench. By right, you must always clear lah, grown plates that are grown. Like the other day, you all grew your plates kan. We ask you all to come the next day itself because they have to be thrown away, discarded. If you leave it, they start growing more and more. You know, cause smell and so on. Okay, it's hazard not good lah because they're pathogenic kan. So they have to be discarded. So what Fleming did when he went on a holiday, he left his plates lying, and he was working on some organism lah, bacteria. And when he returned, although he was untidy, he was smart lah. So he noticed something different on his plate. So he found that the fungi, the part where where how you grow your bacteria on agar plates, there were some parts, so they were moldy already, moldy like food lah. Food you leave a few days, sometimes you see fungi growing kan. But because this was agar plates with bacteria already on it, so when the fungi grew, we found that the area where the fungi grew, or area surrounding it, was clear of the bacteria. The bacteria didn't grow there. Okay, so which later he found out that the bacteria, the fungi, penicillium, was actually producing something to defend itself from the bacteria. So that's how penicillin was discovered. Okay, and then that was 1928, but not until later during I think the first patient that used penicillin was for a woman with a miscarriage. Okay, so this is apparently some advertisement. World War II kan, it was between 1939 and 1944 kan. So during the World War was when penicillin was started to be used highly. Okay, and not only for infection but also for before performing surgery. Even now when we do surgery is performed, the patient is pumped literally pumped with a lot of antibiotics to keep away from infection. So those days they do open surgeries without imagine doing without antibiotics. So the discovery of penicillin was a big importance lah medical in the medical field. Okay, but now a lot of bacteria have developed resistance, but some still are. Some still are sensitive to penicillin. Okay, so still aureus. So more than 90% of Staph aureus are penicillin resistant. Okay, because they can produce penicillinase. Okay, or the beta lactamase. So they have been replaced with penicillinase resistant penicillin like methicillin and oxacillin. Okay, and then we have also methicillin resistant Staph aureus now. Not now, but many years already. So and many other antimicrobial drugs. So vancomycin is used to treat MRSA, and then we also have some resistance to vancomycin. Okay, so in general, prevention of Staph aureus through hand washing to reduce spread because they are flora of nasopharynx and skin. Okay, so that was only our first species for today, Staphylococcus aureus. So next, let's move to Epidermidis and Saprophyticus. Both are coagulase negative, mannitol negative, and they don't hemolyze on blood agar. We saw just now Staph aureus can be beta hemolytic. Okay. Disease that it causes epidermidis can cause endocarditis. Okay, in patients with prostatic heart valves, prostatic hip infection, intravascular catheter infection, cerebrospinal fluid shunt infection, urinary tract infection. So, what can you see in this? What do you think causes all these infection? What virulent factor in Staph epidermidis? You see a lot of prostatic devices, can?
Yeah.
Meaning its ability to produce. Remember bacterial pathogenicity the other day I mentioned? What grows on prostatic devices? What is produced? Remember, like in plastic bottles and all, biofilm. Okay, so that it's because Staph epidermidis ni, although it's a normal flora on our skin, doesn't cause infection on the skin. It if it breaches the skin barrier gets in, and then it can produce biofilm. Okay, like this. Okay. Okay, so infections usually due to patients' own strains that migrate from the skin along tubing or implanted plastic devices. So even and also when taking blood sample, often there may be aseptic techniques need to be taken so that it's not contaminated with the patient's own sample. Okay, that one is for sampling, but in prosthetic device use, these organisms can get into the blood, causing a lot of infections. Okay, it's actually a low virulence organism. Okay, it causes disease primarily in immunocompromised patients and those with intravenous lines, catheters and implants and so on. Okay, so the important virulent factor is biofilm. It's also a major cause of nosocomial infection, Staphylococcus epidermidis. Diagnosis, already mentioned. Treatment, vancomycin, no biocin, and removal of the plastic device. Of course, mainly if you know the cause of it is the plastic device, it has to be removed and changed. Prevention, no drug or vaccine. Next, Saprophyticus. Okay, Saprophyticus is a normal flora of the vagina. Okay, it causes UTI, urinary tract infection, and cystitis in young sexually active females. Okay, so it's also called honeymoon cystitis, honeymooners UTI. Okay, so the female urethra is shorter, and so bacterial transfer is easily can occur in female, okay, causing urinary tract infection. Okay, so during sexual activity, it can cause bacteria can be forced into the uh, urinary tract lah, causing infection, hence those names. Okay, so novobiocin sensitivity test is performed for cultures from urine samples to distinguish from other coagulase negative staphylococci. Okay, have you seen this kind of discs on placed on blood agar plates? Belum kan? I think not yet. Okay, so what these discs are is the disc contains what? Antibiotic. Okay, antibiotic or the drug lah, the antibiotic. The blood agar is growing the bacteria. So like how you did your streaking that day, okay, in antimicrobial susceptibility test, a blood agar plate is filled with the bacteria. Okay, it's like a lawn. You use something like a cotton swab. You dip into a bacterial broth, and then you grow across. In this case, two types of bacteria are being grown lah. One left, one right, and then you place the drug that you want to test, antibiotic, on here. They actually like a filter paper with the drug. Okay, and then you incubate together. Next day when you look at it, you can see whether the bacteria is sensitive or resistant to the antibiotic. Okay, but it's not as straightforward as that. You need to measure actually the diameter. Okay, sometimes this big diameter of this big does not mean it's resistant fully. Sorry, this is sensitive. Okay, does not mean it's there is a certain measurement lah you have to refer to. Okay, you will learn that when you do antimicrobial susceptibility testing with Prof. Nurul. Okay, but here this is just to show that between saprophyticus and epidermidis, because epidermidis was a skin flora kan, and then saprophyticus also now you know that it's from the vagina. So you want to differentiate these two. You put a novobiocin disc, and saprophyticus is novobiocin resistant. That means it can grow in the presence of novobiocin here. Okay, this one is sensitive. Okay, next. Okay, that was our step. Okay, done with our staphylococcus. So we are moving to streptococcus. Okay, so they are gram-positive cocci in chains or pairs. Okay, some are pairs like this. Sepasang. Some are in chains. Okay, we'll see which is which. The normal flora of nose and throat, not on the skin, but nose and throat. So they have a complex nutritional requirement. So blood agar supports their growth. Species that we look at today, or the only species we have under this will be Streptococcus pyogenes, Streptococcus agar-lactiae, Streptococcus pneumoniae, and then a group called Viridans group Streptococci. Okay, and then Enterococcus. Enterococcus is a different genus, but it used to be called strep. Okay, but although it's separate genus, we cluster them together because they are very related. Two ways of classifying Streptococcus is by hemolytic properties on blood agar or serological uncertain grouping. Okay, so blood agar, you can look at when you are given a hemolysis blood agar plate, you must make sure to check if the bacteria is causing hemolysis. Okay, because blood agar contains blood kan, not human blood lah, of course. It's commercially prepared in horse blood or sheep blood. So the blood agar plate you see in the lab, it contains blood. So some bacteria are able to lyse that blood. So I call it hemolysis. But there are three types of lysis. Okay, alpha, beta, and gamma. Okay, so this image here is good to show you that the three all in one plate lah. So you can see here, this is a mistake. This is beta, this is alpha. Okay. Okay, so this is beta hemolysis. Beta hemolysis, it it's not a mistake lah. I just put A for beta hemolysis, B for alpha. Okay, so we like to see what we think. Okay, so beta hemolysis, when you look at the plate, you put under the light, as I said the other day, you can see transparent. You can see your hand behind it because the red blood cells in the agar plate is completely lysed by the bacteria. In alpha hemolysis, it is partial lysis. Okay, as I have given here, partial lysis. Okay, which causes a greenish color from incomplete. Sorry, this is still dark. Okay, incomplete. Alpha is incomplete hemolysis. Hemoglobin is converted to biliverdin, okay, giving that green color when cultured on blood agar. Okay, and then this is gamma no hemolysis. Okay, so in beta there is complete lysis. Okay, and gamma no lysis. Okay, so among the beta hemolytic strains, you can further group them using Lanzaphiel grouping. Okay, so two ways of classifying streptococcus by the hemolytic properties, and next by Lanzaphiel grouping. Lanzaphiel grouping is mainly done for beta hemolytic streptococcus. So you can classify them from A to H and K to B, but most important significant human pathogens are A and B. Have you heard of group A streptococcus, group B streptococcus? Okay, that's because of their Lanzaphiel grouping. Okay, the carbohydrate on their cell wall, it's grouped based on that. Okay, so a latex agglutination test is done to identify that. Okay, the grouping. Okay, so back to our alpha hemolysis. So we saw 6 strains here, kan? Sorry, 5. 1, 2, 3, 4, 5. Okay, so the first two, Streptococcus pneumoniae and viridans group Streptococci, are both alpha hemolytic strains, alpha hemolytic bacteria. But so they can be differentiated by placing an optochin, another antibiotic drug, on the plate. Okay, like I mentioned just now, kan, nomomycin. This is optochin. So optochin, Streptococcus pneumoniae is sensitive to optochin. That means it cannot grow around optochin. So if you see a blood agar plate with alpha hemolysis and a disc placed on it, okay, and it's not growing around it, that means it's optochin sensitive lah. Okay, this one is optochin resistant, which is viridans group Streptococci. Besides that, pneumonia also has a capsule. So it has a positive Quellon reaction. I'll show you that later. Viridans group does not have capsule. Okay. Next, beta hemolysis will be pyogenes and agalactiae. For this, you use bacitracin. Okay, bacitracin disc. So bacitracin disc, bacitracin sensitive, it will be pyogenes. Okay, cannot grow around bacitracin. Agalactiae is resistant. Okay, and then Enterococcus is gamma hemolytic. Okay, so we covered all that 5. Okay, so let's first look at Streptococcus pyogenes. Streptococcus pyogenes is very important in skin, causing various skin infection. Okay. And it's very similar, you'll see it's very similar to Staphylococcus. Actually, you will see one organism very similar to Staphylococcus. Pyogenes is very similar to aureus. Okay, just to confuse you. It's made like that. Okay, so it's already so many to remember, and then they are very similar as well. Okay, so the disease again pyogenic, fast forming, inflammatory and toxigenic. So the pyogenic one, if you think of sore throat, what organism causes virus or bacteria commonly causes sore throat? Virus, virus can commonly, but if it's caused by a bacteria, it is strep lah. Okay, you call it strep throat kan. Then other skin infections, impetigo, erysipelas, cellulitis. We saw some of these in staph just now. Toxigenic disease, scarlet fever, streptococcal toxic shock syndrome. Just now we saw staphylococcal toxic shock syndrome. Now we have streptococcal. And also what causes necrotizing fasciitis. So these are the different skin infections caused by streptococcus pyogenes. Okay, and where they are present, the layer of the dermis or skin layer can cause a different symptoms lah. So this is a pharyngitis. Pharyngitis is sore throat. This is impetigo, erysipelas, cellulitis, necrotizing fasciitis, and scarlet fever here. Okay, and I'm sure you have remembered this from immunology. Immunologic disease, a delayed antibody mediated disease, rheumatic fever kan. In patients who are not treated with antibiotic, the bacteria can cause an immune reaction. Okay, not in all patients, but in some cases. Okay, following untreated strep pyogenes, strep throat lah, sore throat, rheumatic fever, and glomerulonephritis, following sore throat or a skin infection from pyogene. Okay, so you remember this kind from your immunology, right? It's a M protein from the bacteria that can cross-react with um, the heart tissue, and then here in acute glomerulonephritis, this antigen antibody complex is deposited on the kidney glomerular basement membrane, okay, causing destruction. So it's acute glomerulonephritis. Okay, how is it transmitted? The habitat now is the skin and oropharynx in small amount. Okay, so there is some strep on skin, but mainly it's staph on skin. So it's transmitted via respiratory droplet. Okay, I won't go through this much. So this is M protein, capsule, enzymes. Okay, you can produce hyaluronidase, streptokinase, DNase, and 5 important toxins and hemolysins. So the hemolysins that lyse red blood cells are streptolysin O, streptolysin S. So both of these can cause the beta hemolysis, or you see on blood agar. Okay, so one for the upper part of the blood agar, one within. Okay, you don't need to know that, but it's a streptolysin lah. Okay, that is the that gives you the beta hemolysis pattern. And this anti-streptolysin O can be used to diagnose rheumatic fever. Okay. And then 3 other toxins: erythrogenic toxin, which is a super antigen similar to staph. Okay, that's responsible for scarlet fever. Okay, so this when we so bacterial transfer of genetic material if you remember, transduction can cause some virulence factor to be transferred. So only strep pyogenes that has been lysogenized by a bacteriophage will carry this toxin Okay, next, pyogenic exotoxin A. Okay, so this one responsible for the toxic shock syndrome in pyogenes, strep pyogenes. Exotoxin A in necrotizing fasciitis. How is it diagnosed? All this mentioned earlier, right? I think I mentioned all of that. Treatment, penicillin is still effective, okay, for streptococcus pyogenes, or generally for streptococcus. Okay, so you would see that a lot of similarities between staph and strep in terms of symptoms, and also they are both cocci under the microscope. Okay, so that's why it's very important to differentiate these two. Okay, because it's not the same drug to treat them. You need the streptococcus is still penicillin sensitive, so it can be used for staphylococcus. You need to do antibiotic, antimicrobial susceptibility test to determine. Okay, prompt treatment of streptococcal pharyngitis to prevent rheumatic fever, and also as prophylaxis in patients who have had rheumatic fever before, okay, to prevent reinfection. Okay, next, agalactiae. Okay, so we saw they are basitracin resistant. Mainly causes neonatal meningitis and sepsis. They cause sepsis, pneumonia, postpartum endometritis in adults. Okay, how is it transmitted? The habitat is the vagina. Okay, just now we saw Staphylococcus, kan, in Staphylococcus, habitat vagina can cause UTI, right? In women, and this one is agalactiae. Also causes, harbored by some women, okay, not all, in the vagina. The transmission can occur during birth and can cause meningitis lah in the baby. Yeah, if so, ampicillin, but to prevent, ampicillin can be provided to the pregnant women who test positive for Streptococcus agalactiae. But then this is not a routine test done. Okay, you can ask for a test to determine. Okay, characteristic. It has a lancet shape cocci in pair. It is diplococci with a lancet shape. Lancet shape is something like this, the shape like a old churches and even mosques kind of the windows had like a shape like that. So you see two shapes like that. It's usually a strep pneumoniae. It's a diplococci. Sensitive to bile, so it can solubilize in bile, and it's also sensitive to octochin, as I mentioned earlier, contrast to viridans. It has a prominent polysaccharide capsule, so you see it a bit like braille lah on the plate. Okay, and it's one of the 3 classical encapsulated pyogenic bacteria. So it has capsule, just like Neisseria meningitidis and Haemophilus influenzae. And all 3 of these, they cause, one similarity in these organisms are they cause meningitis. Okay, because for meningitis to happen, bacteria need to survive in blood and then cross the blood-brain barrier, kan? So this bacteria, all 3 have capsule. Okay, the capsule allows them to do that, lah. Okay, antiphagocytic. So what does Streptococcus pneumoniae cause? As the name suggests, the most common cause of bacterial pneumonia, besides meningitis, sepsis, otitis media in children, swim and all, swimming. Okay, how is it transmitted? The habitat is the human oropharynx, transmission via respiratory droplet. Okay, so capsule, I mentioned this before, it has 90 distinct capsular serotypes. I hope you understand what this means. So it's just that the different types of polysaccharide that where the host can produce anti-serum lah, antibody towards those 90 different capsular serotypes. So it helps in serotyping, find out what strain it is. 23 types cause most of the infection. So okay, the rest we have seen. So this is Quellem reaction. Okay, so what happens here is we add a pneumococcal antisera. Okay, antisera for specific for Streptococcus pneumoniae, and then to see, of course, you need to add color, so methylene blue, and the diluted bacterial suspension. So the reaction between the antibody and the carbohydrate in the capsule causes some reaction, making the capsule looking swollen. Okay, so that is Quellem lah. So if you see this, it means Quellem positive. And also you can see here the bacteria is present in pairs, right? Like what I said. Okay, Streptococcus pneumoniae are, I think the only gram-positive cocci you see in pairs will be Streptococcus pneumoniae. Okay, gram-negative you will see Neisseria, Neisseria meningitis also present in pairs. This one, but gram-negative lah, pink. Okay, so these are all small little clues when you see a specimen in OSP. If you see gram-positive cocci in pairs, it's definitely Streptococcus pneumoniae lah. Okay, but you have other tests to be sure. Okay, all the alpha hemolysis and so on. Okay, so it produces, besides capsule, produce autolysin, pneumolysin, IgA protease, teichoic acids, and hyaluronidase. Diagnosis, all mentioned, I think, yeah. Treatment, penicillin. Vaccination, the vaccination targets those capsular antigens. Okay, so it's very antigenic. So there's two types of vaccines. I think you will see this more clearly in Prof. Ilina's lecture in the next semester on bacterial borne respiratory infections. Okay, so for Hajj, during Hajj, it's recommended, isn't it, for the vaccination for elderly. Okay, so the difference between these two are how many they target. Okay, how many different antigen target. Okay, and then now viridans group streptococci. Okay, so they are a group of streptococci, mainly mutants, Salivarius, Anginosus, Mitis, Sanguinis, and Bovis. Okay, just remember viridans group and remember lah, there is a few here, mutants, Salivarius, contoh. So they are uncapsulated, cause endocarditis and dental caries. So in Staphylococcus, we saw epidermidis causing biofilm present in prosthetic devices, kan? So Streptococcus pun must have lah counterpart. Okay, so this is viridans. Okay, so it's a normal oral flora, can enter bloodstream during a dental procedure. Okay, during dental procedure, there's some wounds and all, kan? Cause blood, you see blood coming and all that. So sometimes the bacteria present in the oral area can get into the bloodstream and cause this endocarditis lah. Okay, so that's why okay, so they have the ability to synthesize dextran or glycocalyx, allows them to adhere to pre-existing fibrin platelet at damaged heart valves. Okay, for dental caries, the dextran can stick to teeth, resulting in biofilm known as plaque. So diagnosis, I think we have seen all this. They are resistant to bile and octocin as compared to pneumonia tadi. Treatment with penicillin, and to prevent endocarditis in patients with damaged or prosthetic heart valves who undergo dental procedures, for penicillin has to be given as a prophylaxis before the dental procedure to avoid this infection. Okay, because it's very hazardous. Okay, so dentists, they usually need to ask before a dental procedure. They must ask if they have all these conditions. Next, Enterococcus. Okay, this is the last slide. Okay, so I think this is a record breaking that I finished it almost fine. Enterococcus is non-hemolytic, unencapsulated, bile insensitive. Although it is not beta hemolytic, I told you Lancifil grouping is done only on beta hemolytic streptococcus, but it used to be a strep and it happens to be group D, so it's called group D Lancifil. Okay, Okay, so it's a way of classifying it. There are also normal colon flora in the gut. So in the feces lah, they release in the feces. They are able to adhere to human epithelial cells and can cause, they seldom cause disease, but when introduced to other body parts during surgery and so on, then they can cause an infection, serious infection. And there is a big problem with this vancomycin resistant strains. Okay, any strain that develops antibiotic resistance is difficult to manage and becomes a problem. Okay, it is difficult to treat. So penicillin or vancomycin in combination with an aminoglycoside. So it can cause urinary tract infection, wound infection during surgery, bacteremia, and subacute endocarditis. Okay, so that was Enterococcus. Okay, sorry, that was not the last slide. This one is the last slide. This chart is a summary lah. Okay, a summary of how to classify these bacteria, gram-positive bacteria. So cocci, okay, gram-positive cocci. We do a catalyst test. All staphylococcus are catalyst positive, and staphylococcus ni can be further identified through coagulase test. Okay, they are all in clusters. Staphyl, so coagulase test differentiate between aureus and coagulase negative staphylococcus.